Hello, I'm Jean Riddell, and this is Immunohematology Review. My references are the Clinical Laboratory Science Review, A Bottom Line Approach, and Denise Harmoning's Modern Blood Banking and Transfusion Practices. Immunohematology Review Part 3. This covers other blood groups, antibody detection, and anti-human globulin. Other blood groups. In the Lewis blood group, a person with which Lewis phenotype will develop antibodies. Someone who is Lewis A negative, B negative. A woman becomes Lewis A negative, B negative during pregnancy and makes antibodies to Lewis antigens. If the baby inherits the Lewis gene, can they be affected by hemolytic disease of the fetus and newborn? The answer is no, because antigens are not on fetal cells and antibodies are IgM. Lewis antigens are formed in plasma and then absorbed onto the red blood cells. What substance is converted to Lewis antigens? Type 1 precursor substance. What antigen is created? I have type 1 precursor substance. I have the capital SE gene. I have the Lewis gene. What antigen do I get? I get Lewis B. What antigen is created? I have type 1 precursor substance. I have the Lewis gene product. And what antigen do I get? I get Lewis A. What antigen is weak or absent on fetal cells but is present on adult cells? The capital I antigen. What antigen is found on infant cells but is converted to a branched carbohydrate chain as they grow up? The little I antigen. What cold reacting IgM antibody will react with all adult cells but not with fetal cells? Anti-capital I. What cold reacting IgM antibodies can be neutralized by hydatid cis fluid? Anti-P1. What IgG autoantibody is known as the Doneth-Landsteiner antibody? Anti-P. The Doneth-Landsteiner antibody causes what condition? Paroxysmal cold hemoglobinuria. Antibodies to which blood group antigens are cold, IgM, no hemolytic disease of the fetus and newborn, show dosage. The antigens are destroyed by enzymes and found on glycophorin A. MNN. Antibodies are to which blood group? It's warm, IgG, hemolytic disease of the fetus and newborn, and has dosage. The antigens are destroyed by enzymes and found on glycophorin B. Big S and little s. The most immunogenic antigen after the Rh antigens and most likely antibody to be found in the blood bank is? So the most immunogenic antigen is big K or KEL, and anti-KEL is the antibody. Which KEL blood group system antigens are considered low incidence? KEL, JSA, and KPA. Notice that in each one, it's the first one that's low incidence and the second one that is high incidence. So KEL is low incidence, Solano is high incidence, JSA is low incidence, JSB is high incidence, KPA is low incidence, KPB is high incidence. Which KEL blood group system antigens are considered high incidence? Solano, JSB, and KPB. Here are the codominant alleles in the KID blood group system: JKA and JKB. A null phenotype in what blood group system causes the myeloid phenotype? Kel. Which blood group are we talking about? Antibodies are IgG, enhanced with enzymes, titers rise and fall rapidly. They can cause delayed hemolytic transfusion reactions, dosage, and bind complement. Antigens are not very immunogenic. This is the KID blood group. So binding complement is like the key thing on this one, binding complement. A person who makes anti-U is most likely from what demographic group? And what antigens would they also be lacking? So they would most likely be African American. They would be lacking U, big S, and little s. The majority of African Americans are what Duffy type and why? Duffy A negative, B negative, because it's protective against Plasmodium vivax. Plasmodium vivax is an intracellular red cell parasite that causes what disease? Malaria. What blood group? Antibodies are IgG, they show dosage. Antigens are destroyed by enzymes, and it has 3 alleles. Duffy, because you have Duffy A, Duffy B, and just FY, which is what the people that have Duffy A negative, B negative have. Okay, we have a father that is Duffy A positive, B negative, a mother that is Duffy A negative, B positive, and a child that is Duffy A negative, B positive. Is this possible that those are that child's parents? So if the father is A positive, B negative, that means he has 2 copies. He could have 1 copy of Duffy A and then a Duffy, the FY, and the mother could be homozygous for Duffy B, or she could be also FY and FYB. So is it possible? Yes. So the father could be Duffy A and FY, and the child could be Duffy B and FY. Father is Kel negative and Solano positive, and the mother is Kel negative and Solano positive. The child is Kel positive and Solano positive. Is this possible? How? No. This is a direct exclusion because the child has an antigen that neither parent has. The father is type AB and the mother is type A. The child is type O. Is this possible? How? No, this is a direct exclusion because the alleged father demonstrates 2 different markers in the same system and the child does not demonstrate either. Father is JKA positive, B negative, and the mother is JKA positive, B positive. The child's type is JKA negative, B positive. Is this possible and how? No, this is an indirect exclusion because the child would have to get JKB from both parents and the father does not have JKB to give to the child. In this example, the father is Kel positive, Solano negative. The mother is Kel negative, Solano positive. The child is Kel negative, Solano positive. Is this possible and how? No, this is an indirect exclusion. The father has 2 Kel genes and the child did not inherit one. A 2-cell antibody screen set would have to be which 2 Rh types? Think about it. We need cells that are homozygous, and they need to cover all of the Rh antigens. So we would have, for screen cell 1 would be R1, and screen cell 2 would be R2. To avoid reactions with ABO antibodies, all screen and panel cells must be which ABO type? The answer is type O. Antibody detection. An antibody screen and ID panel are considered what type of test? An indirect antiglobulin test. During an indirect antiglobulin test, the tech performs only 2 washes to speed things up, failing to remove unbound proteins. What would we see in their test results? The check cells would not agglutinate and the test would be invalid. What blood type are check cells and what are they coated with? They are type O positive and they're coated with anti-D. While performing an antibody panel, some of the panel cells hemolyzed after the 37 degrees Celsius incubation. How would we categorize this reaction? It is a positive reaction. Antibody detection. While performing an antibody panel, after the AHG is added, some cells are positive and some are negative. What is the next step? Add check cells to all the negative cells. I completed 2 panels. One is enzyme treated and the other is not. Otherwise, they're the same cells. Some of the reactions went away on the enzyme treated panel, but not all. What does this tell me? There is an antibody to an antigen that is destroyed by enzymes, and there is an antibody to an antigen that isn't destroyed by enzymes. What characteristics do the antigens MN, capital S, lowercase s, and Duffy have in common? They are destroyed by enzymes. Remember, it's the antigens that are destroyed by the enzymes when the cells are treated. What characteristics do antibodies to MN, capital I, lowercase I, P1, and Lewis have in common? They are IgM or cold reacting. What characteristics do antibodies to KID, RH, Lewis, I, and P have in common? They are all enhanced by enzymes. What characteristics do antibodies to RH, Kell, KID, and Duffy have in common? They are IgG antibodies. What characteristics do antibodies to RH, KID, Duffy, and MNS have in common? Dosage. Anti-human globulin. What type of AHG is derived from animals and will target multiple epitopes? Polyclonal. What type of AHG is a mixture of antibodies to IgG and complement components? Polyspecific. What type of AHG is derived from hybridoma technology targeting a single epitope? Monoclonal. What type of AHG contains an antibody to either IgG or complement, but not both? Monospecific. If I wanted to avoid detecting cold complement binding clinically insignificant antibodies, which AHG reagent would I use? Monospecific anti-IgG. If I wanted to first detect a positive DAT and then characterize it by the type of substance that's attached to the cells, which AHG reagent would I use and in which order? First, I would use polyspecific AHG, and then if it was positive, I would use a monospecific anti-IgG and a monospecific anti-C3b in separate tubes to determine which is the which is coding the cells. If a patient had hemolytic disease of the fetus and newborn, or a transfusion reaction, what type of positive DAT would we expect? IgG coded. If a patient had cold hemagglutinin disease or paroxysmal cold hemoglobinuria, what type of positive DAT would we expect? Complement coded. If a patient had warm autoimmune hemolytic anemia or mixed type autoimmune hemolytic anemia, what type of positive DAT would we expect? IgG and or complement coded. Detecting antibodies, a procedure where we break antigen-antibody bonds to release antibodies into solution is called elution. A Louis freeze-thaw elution is performed to remove which of the following? ABO antibodies or complement? ABO antibodies. An acid or organic solvent elution is performed to remove which of the following? Warm reacting antibodies, allo or auto, or complement? Warm reacting antibodies. When we perform an elution, the solution containing the antibody is known as what? An eluate. Performing an eluate, the last wash should show no reactivity with reagent cells. If there is still reactivity, what step would you take to remove antibodies in the solution? You would wash again. What is the procedure that uses soluble antigen to bind with antibody to inhibit a reaction with red blood cells? Allows detection of alloantibodies masked by Lewis substances in saliva, P1 substances in hydatid cyst fluid and pigeon egg whites, or SDA substance abundant in urine. This is neutralization. The process uses sulfhydryl reagents to destroy antigens or antibodies, uses chloroquine diphosphate to remove IgG from cells for phenotyping, or uses acid glycine EDTA to dissociate antibodies from red cells is called what? Inactivation. The process that is used to separate multiple antibodies by removing one antibody by reacting it with antigen-positive cells can be done with autologous cells for autoantibodies and with allogeneic cells for alloantibodies is called what? Adsorption. Thank you for listening. Please continue to part 4 of the Immunohematology review. Remember to share, like, and subscribe to my channel.